Hi, I'm Mrs. Sloan, and this is a video for my honors bio students, and this is part 2 of our chapter on evolution. Part 1 was about the evidence for evolution, and now part 2 is going to be on the process of evolution, how that would come about. So let me make myself a little bit smaller. There we go, and get in presentation mode. And put this away. Okay, here we go. So we're going to be talking about the process of evolution. Now, the notes are down in the descriptor for the video. Column 1 is a scaffolding of notes, which I will help you fill in. And column 2, I encourage you to put in pictures or images that help you remember that information. All right, so here we go. The first thing we need to know is, let's start with this frog. Why might this frog, who is brightly colored, why might it be those colors? Is it possible that this frog is poisonous and you want to advertise the fact that you're poisonous so that other organisms don't eat you, right? And they learn quickly, if you eat the yellow ones, then you get sick. So keep in mind a couple of terms I want to introduce you to, even though we haven't in my class yet discussed genetics. I want to teach you these two words, genotype and phenotype. Phenotype is the appearance of this frog with its blue underbelly and its brown and white here on its back. That's the appearance. Its genotype are the actual DNA, alleles on that DNA that coded for those particular traits. So evolution by definition is when you have a change in traits over time, and that has to mean that you have a change in the alleles, the DNA that codes for those traits in a population over a time. A single frog cannot evolve. It would have to be a population. So evolution is, and this is where we start on the notes, evolution is about a change in traits that are governed by alleles. So traits is the phenotype, what it looks like. Alleles is the genotype, what codes for that. So evolution is about a change in traits governed by alleles in a population, which is a group of a single species living together in the same geographic area. Specifically, the change in allele frequencies in a population over time. So let's look at the factors that would contribute to that. Let me put myself over here. Here we go. So evolution is a result of 4 factors. You have to have potential to increase your numbers of one of those traits. You need to have variations in your DNA, competition for resources, and know that the best adapted wins usually and survives. That's called differential reproduction. So let's look here. First of all, you could have a variation in beetles. Okay, you could have some brown beetles, you could have some green beetles. If this population of beetles is living on a brown tree, the brown ones are going to be better adapted because they're going to be able to hide better. And the green ones are going to be more visible. And so the birds are probably going to eat the green ones more. So you're going to have an increased reproduction of the brown ones. And over time, you'll have more and more brown beetles and less green beetles. That is evolution. Evolution does not have to be a change in species. You can have an evolution within a species, and that is referred to as microevolution. So these could be one species of beetles just with a variation in their color, just like we have variations in hair color and eye color, right? We're still the same species. But you could have differential selection or reproduction of one of those species if they are more adapted. So on your notes, results from 4 factors: the potential for a species to increase in numbers, the heritable, meaning it's in your DNA, the heritable genetic variation of individuals in a species due to a mutation or sexual reproduction. We'll get more into that later when you and I study meiosis together. C, competition for limited resources. And then D, the proliferation of those organisms that are better able to survive and reproduce in the environment. So in this case, the brown ones are better able to survive and reproduce because they're able to hide. All right. Now let's look at the 2 types of evolution that I told you about. Okay, so microevolution is within a population, a change within a population. For instance, humans here in the United States have gotten taller and taller and taller. And it could be due to our eating habits. It could be due to medicine. but we have definitely gotten taller here in the United States on average. Macroevolution would be if changing to a whole new species where you're reproductively isolated. So types of evolution, microevolution is within a population, and macroevolution is large scale, forming a new species. So I want to give you the most well-known microevolution example, change within a species, similar to what I just showed you about those beetles and the birds. This is called industrial melanism. And pre-industrial revolution, you have two kinds of moths. One species with two different kinds of colors. You had the black version of this species of moth, and you had the lighter or gray version of this species of moth. Now, when there was no pollution, obviously the lighter colored moth, let me get a pointer, obviously the lighter colored moth was more well hidden. And so the black moths would get eaten more readily, their numbers would go down, and so they wouldn't have the chance to reproduce. So when you looked at those two colors within a population, the lighter colored moth would be more prevalent than the darker colored moth. Post-industrial revolution, okay, you had all this soot that was on the trees. Now, within that same species, you were more adapted if you were the darker colored moth because you blended in with the soot. And so their numbers increased. This was an example of microevolution because we didn't change to a new species. These two could still interbreed, but the frequency, the numbers changed, and you had more of the black than you had the lighter one. Okay, that was a change in allele frequencies within their population. So when you, I have another example for you. I forgot about this. Sorry, here we go. So this is the yellow-bellied three-toed skink. Now, the more common version, they lay eggs, and then those eggs hatch. But what they have found is those that have migrated to a colder climate are keeping their eggs within them longer. And so there are actually some of this species of the yellow-bellied three-toed skink are giving live birth over time. And here's the article. If you really want it, you can freeze on here and take a look at this article. It was from National Geographic. And the idea is that right now, they still interbreed with each other, the ones that give live birth and those that lay eggs. But if over time, the ones that have live birth, because their babies are protected longer within them, right? If they only mate with those that also give live birth, then this will transition from microevolution into macroevolution. All right. So now, there is a great video. I'm going to show this to my class during class, but I encourage you, this is from Bozeman Science. It's just search it, The 5 Fingers of Evolution. It's a great video that will explain what I'm going to talk about right now. All right, so first thing we need to know is a population is all the members of a single species occupying a certain area at the same time. So they could all interbreed with each other, okay? Now, it's really hard to talk about humans and populations because you have the ability to travel all over the place. But I'm going to try to simplify this a little bit, give you a little more genetics. When through sexual reproduction, sperm will carry, for humans, 23 chromosomes, and the egg will have 23 chromosomes in it. They're not all drawn here. And then through fertilization, you have what's called a diploid zygote. For you and I, that's 46 chromosomes total. So you get a chromosome 1 from mom, and you get a chromosome 1 from dad. So you have 2 copies of information. Now, dad could have brown eyes, and mom over here could have blue eyes. The mom, let's say she gives the blue-eyed allele and the dad gives the brown-eyed allele. Brown eyes are dominant over blue eyes, so this little girl would have brown eyes. So variation in alleles, brown-blue, but brown wins over blue, and this little girl would express the brown-eyed trait. So when we look at individuals who have brown eyes, remember, phenotype is what you look like, and genotype are the alleles that code for it. This brown-eyed person could be big B, 2 brown-eyed alleles, receiving a brown-eyed allele from mother and a brown-eyed allele from father, and they would have brown eyes. Or this person with brown eyes could be a carrier for the blue-eyed trait. We wouldn't know, right? Because as long as they have one brown-eyed allele, they will have brown eyes, regardless if the second allele is blue-eyed. Now, we're going to talk all about that in genetics, and that's coming up in a different unit, but right now I just needed to introduce that to you. This blue-eyed person, the only way they could have blue eyes is if they were given a blue-eyed allele from both parents. Now, could their parents have had brown eyes? Yes, both parents could have brown eyes, and they could have just given the blue-eyed allele to their offspring in order to give them blue eyes. All right, two alleles here, both of them blue. Here, we're not sure. We just know one of their two alleles is the brown-eyed trait. So let's talk about now some wild boars. And let's now make these alleles, we'll still use Bs just for a little bit here. We will say that if you have the big B allele, you have bristly hair. And if the little b allele codes for smooth hair. So any individual who has at least one big B here, remember they have two Bs, one from mom, one from dad, right? Any individual that has a big B, this would be a bristle-eyed, bristle-haired. This one, because they have two little Bs, would show the recessive trait, which is smooth hair. This one has bristle, smooth, bristle, and bristle. Now, there are names for these. When it's homozygote, homo means same. So in this case, big B is homozygote because both of the alleles are big Bs. Okay, this one right here is homozygote because they have two little b's. They got the same from each parent. This individual here with a big B and a little b, that would be hetero or different zygote, heterozygote. They are a big B and a little b. So we could count up and find out in this whole population of wild boars, what is the frequency of each of those alleles. So I could count up all the alleles I have here, right? I have 1, 2, 3, 4, 5, 6, 7 boars in this population. Each has two alleles, so that means a total of 14 alleles, right? If we counted these up, you would count a total of 14. So if I look here, the dominant allele, when you do genetics and do population studies, the dominant allele is referred to as P and the recessive allele is referred to as Q. That's just a standard regardless of the trait. So for instance here, if I counted up the P or the big B allele, the dominant allele, I have 1, 2, 3, 4, 5, 6, 7. 7 of the 14 total alleles in this population are big B. So the P allele frequency is 0.5 or 50%. Well, if the dominant allele is 50%, then you know the recessive alleles got to be 50% too, right? Because those are the only alleles you have. For instance, if I said the brown-eyed allele was 0.75 in the population of interbreeding people, then the recessive blue-eyed allele would have to be 0.25, right? Because that equals 1. So this, when you look at these numbers, this is part of population genetics. And you need to understand that basically what we're going to learn, cutting to the chase here, is that if allele frequencies change, if this changes instead of 0.5 and 0.5, if it changes to 0.6 and then what, 0.4, then allele frequencies have changed, and by definition, that is evolution. It doesn't matter if you change species or not. It doesn't matter if it's micro or macroevolution or not. If the allele frequencies change, then evolution is occurring. So if you look underneath population genetics on your notes, it's defined as the study of the changes in the genetic makeup, the study of the changes in the genetic makeup of a population in their gene pool, The study of the changes in the genetic makeup of a population in their gene pool. What is a gene pool? So this is your little analogy here of a gene pool. It's all the alleles in the population, So if each of these boars took their two alleles and threw them into a pile, that would be your gene pool. And right now, by counting them up, we know 50% in the gene pool are big B and 50% are little b. All right, this leads to these two equations about allele frequencies and genotypic frequencies. Okay, now allele frequencies, we know that the big B allele was 0.5. So we know They all have to add up to 100%, right? So if the big B was 0.5, then the little b had to be 0.52. Just like if I said if the big B allele was 0.6, then the little b allele would be 0.4. You can do the math on that. That's within the gene pool. So this represents right here allele frequencies in our gene pool. Now, genotypic frequencies is this. Within that population, if you're going to make a baby, this is your gene pool in your population. If you're going to make a baby, remember a baby's going to have two alleles. If I reach in there, what are the odds that I create a baby with two big B's? What are the odds I create a baby with big B, little b? What are the odds I create a baby with little b, little b? Well, you just do the math on that. You do the odds, right? So homozygous dominant would be like that big B, right? So what are the odds of you pulling it out? Well, you know the frequency of the big B allele, or what we're calling P, is 0.5. If we're going to pull out another one, it's 0.5. You just multiply that together. 0.5 times 0.5. I had a 25% chance of creating a homozygous dominant allele with that allele frequency. You just do the math. So there's a little bit more on the heterozygotes. Those are the ones, remember I told you like a brown-eyed person could be brown or brown blue, carrying that blue-eyed allele that you didn't see. Those are your heterozygotes here. So you could either pull out a P and a Q or a Q and a P. That's why we multiply it by 2. And that's why, right, 0.5 times 0.5 is 25 times 2. You had a 50% chance of reaching into your gene pool and pulling out a P and a Q or a Q and a P in that way. And then Q squared, there was a 0.5, a 25% chance that you would get a homozygous recessive boar. All right. Now, here, okay, you can predict using P squared plus 2PQ plus Q squared equals 1, the frequency. And I did that for you right here. Okay, I just wanted to write it out mathematically for you. P equals the dominant allele frequency, or big B. Q is the recessive allele frequency, or little b. Okay, remember we counted up that we had 7 and 7, or 50%. So then P squared, 25% of those boars, if you wanted to predict, 25% of the boars would be homozygous dominant, 50% would be heterozygotes, and 25% would be homozygous recessive. All right, so now let's try it with another scenario, okay? If I was looking at pea plants, tall pea plants are dominant over short pea plants. So if I have a whole field, a population of pea plants, and 36% are tall, and being short is recessive, could I figure out my gene pool that it came from, right? When I say 36% are tall, they've either got to be PP or PQ, right? Because P is dominant over Q. So what is the right down here? Remember, tall would be P squared, or your 2PQ'd part of that equation, right? So what is the frequency of the tall allele? I'm trying to figure out what's in their gene pool if this many are tall, right? And how many would be carriers. So let's do the math on that, okay? And I know this might be, let me see if I can get myself over here. So, okay, no, apparently not. Okay, so let me explain the math on this, and you can rewatch this if you need to, and plus I'll be talking about it in class, all right? So here are our two equations. This right here, P plus Q equals 1, that's your gene pool, and then this comes about from reaching into the gene pool and making some babies. This is your genotypes that are out there. All right, so if 36% are tall, those equal the P squareds, the homozygous tall, and the carrier talls, right? But if 36% are tall, that means 64% have to be short, the recessive trait. That would be your Q squareds right here, right? If this many here, 36% are tall, both of these genotypes would be tall, this would be your short, 64% are short. Well, you can do the math on that. If Q squared equals 0.64, then if you take the square root of each side, then you know Q equals 0.8. That means in the gene pool, 80% of the alleles in our gene pool are little q's, okay? That means what's left then? If 80% are Q, then since P plus Q equals 1, what would P be? P would be 0.2. That is the frequency of the tall allele. So for this population, okay, 0.8 is the recessive allele. 0.2 is the dominant allele. What did I tell you? If those allele frequencies change in any way, then what is occurring? Evolution, exactly. All right, so let's figure out how many were carriers. Remember, to be a carrier, those are the two PQs. So 2 times 0.2 times 0.8, that means 32% of those tall plants were actually carriers that were masking the short allele. And then from that, I could figure out how many are P squared, right? I could do it a couple of different ways. Remember, 36% total were tall, but now that we know that 32% of them were the carriers, that leaves 4%. Or so 36 minus 32 is 4%. Or I could just figure it out because 0.2 was the frequency of the tall allele. 0.2 times 0.2, right, is going to be 0.04 or 4%. Okay, that is, again, looking at allele frequencies. And we use that to talk about evolution. Let's give another scenario. Okay, so in this case, if purple flowers are recessive, what is the allele frequency in this population? Well, if you count all these up, I believe I have 12 flowers here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. How many of these 12 flowers are purple? Well, 1, 2, 3, 4. So 4 of the 12 are showing the recessive trait. And that's what I have to use. Because remember, these yellow ones could be PP or they could be PQ. We wouldn't know because yellow is dominant over purple. So any of these yellow flowers could be hiding the purple trait. So that's why I need to use the recessive trait, because that's the only clean number I've got. Because the only way to be purple is if you're Q squared. So 4 out of the 12 show that recessive trait. So when I look at that, again, let me move myself over here. So the math on that, if 12 flowers total and 4 are purple, that means 4 out of the 12 have the recessive trait, or 33%. So that means my Q squared is 33%. So when I take the square root, it's really 0.3333, but when I take the square root, that's 0.57. Then in my gene pool that created that 33% purple flower, the gene pool that it came from, 0.57 alleles in that gene pool are the recessive trait, and 0.43 then would be the dominant allele frequency of the yellow allele. All right, so we can go to your notes again for a minute. Okay, so we said allele frequencies. The gene pool is all the alleles in the population. The gene pool, if you want to add to that, is P plus Q equals 1. That is your gene pool number, all right? The allele frequency is the percentage of the gene pool for one allele versus another. All the allele frequencies should add up to 100% or 1. And now let me go one step further. If you were given, this would be super easy. If you were just given the allele frequency, either P or Q, you could predict the percentage of each genotype. How many homozygous dominants, heterozygotes, or homozygous recessive? So if P equals 0.6, then what does Q have to be equal to? Did you say 0.4? I hope you did. If P is 0.6, then Q equals 0.4 because P plus Q equals 1. That is our gene pool. That means if I reach in here 60% of the time, I would predict I would pull out the P or dominant allele, and 40% of the time I predict I would pull out the recessive allele. So now I could figure out each genotype using this equation right here. So 0.6, 0.6, you multiply that together, that's 36%. I would predict in my population, if that's my gene pool, 0.6 and 0.4, I would predict 36% would be the homozygous dominant trait. Then I could pull out a 0.6 and 0.4, or I could pull out a 0.4 and a 0.6. That's how we say 2 times that. So 48% are heterozygous dominant, or 2 PQs. And then Q squared, that would be 0.4 times 0.4, or 16%. All right? So look at this right here about gene frequencies. Now we're going to learn pretty soon about Punnett squares in genetics. So basically what you did is you took, here's my gene pool for my eggs, could either be 0.6 or 0.4, right? Whether the dominant or recessive allele. Same thing for the sperm in my population. So I had a 36% chance of getting a homozygous dominant, right? 0.6 times 0.4 or 0.6 times 0.4, either way, this is my 48% being heterozygotes, that's your 2pq, and 16% little q, or in this case, the recessive trait, little l. So population genetics is the study of the changes in the genetic makeup in a population within their gene pool, the changes. And if the allele frequency in that population changes, then evolution is occurring. For instance, if this big L right here changed from 0.6 to 0.7, could you do the math on that? You might pause the video for a second and figure out all the frequencies right here. If this changed to 0.7, how does that alter everything? So hit pause if you want to. If not, here you go. Okay, so if we did the math on that, if this was 0.7, then Q would equal 0.3 because 0.7 plus 0.3 equals 1, right? Then you could figure it out. 49% would be homozygous dominant, 42% would be heterozygotes, and 9% would be homozygous recessive. So that's letter C on your notes. If the allele frequencies for a particular trait remain unchanged, P and Q values remain the same generation after generation, then that trait is not changing and not evolving. If you can get these allele frequencies to stay at 0.7 and 0.3. If you can get it to stay there, then no evolution is occurring. And it's said to be in Hardy-Weinberg equilibrium. Now, that's pretty difficult to keep those allele frequencies the same. You have to meet some conditions in order to do that. Okay, so we're going to talk about the Hardy-Weinberg law. If you can freeze allele frequencies, then you can stop evolution. If not, then evolution will happen. So let's look at all the conditions to keep those allele frequencies exactly the same. So number one, okay, you need to have no mutations because you could mutate, right, from P to Q or Q to P, from big L to little L or little L to big L. So you would have to stop all mutations. Number one, no mutations. Number two, you need to not have any gene flow. You can't have any new alleles coming in to your population or any alleles leaving. So number two, no gene flow. The third condition you have to meet is that all your populations have to be large. Every single population has to be large. Otherwise, you're more subject to chance changing your population and the numbers within your populations and therefore your allele frequencies. So number three, you have to have large populations. Number four, you have to have absolute random mating. No one trait can be better than another trait. And it's always random who you mate with. Now, do you think that's pretty normal? Probably not, right? Usually there's some sort of selection or some trait that you're going for in order to who you're going to mate with. But in order to freeze allele frequencies, random mating. And number five, no selection. Nothing making you better adapted to your environment than anything else. And I'm going to take you back to that frog I started out with. Remember all the colors on that frog? That frog is a poisonous frog. He's advertising that he is poisonous so that there is a learning curve of those organisms that might hunt them. And they're like, don't eat the yellow ones or don't eat the bright blue ones because they're going to make you sick. Now take a look at this, right? Here's a cobra. And on the back of this cobra's face, it looks like there's a face. Do you think he's ever seen his face? Probably not. How did this get here? Well, potentially, right? Those organisms might want to, if they were going to hunt this snake, they would want to sneak up on this snake. So they would go around from the other side and approach from this side. Little do they know, this is the actual mouth and face, bite, death, that whole drill. So those snakes that had what looks like a face were more likely to survive and reproduce because their predators were confused. So that would be a form of selection. If you don't want the allele frequencies to change, then you cannot have selection. And how likely is that? No selection. So that's your fifth one. So the assumptions of Hardy-Weinberg equilibrium, it's just a different order, but the same thing. Okay, you can't have any selection. No one is better than the other. There's no mutations, there's no migration. You always have to have a large population, and you have to have random mating in order for that to succeed. And then right there underneath your notes, you have the two Hardy-Weinberg equilibrium equations. I already gave those to you right there in your notes. Okay. And since it is very difficult to meet all of those conditions, therefore evolution is probably happening because it is difficult to meet all of those conditions. So since it is difficult not to violate these conditions in natural populations, alleles must be changing, and therefore evolution must be occurring. Alleles must be changing, and therefore evolution must be occurring. All right, so let's look at those agents one more time in a little more detail. So we are under microevolution and Hardy-Weinberg equilibrium. The Hardy-Weinberg principle only applies if the population remains unchanging. The following items can cause change, which would therefore cause evolution. So number one was mutation, change to the DNA sequence. These mutations can serve as a source of variation. By the way, back to those eye colors, those of you who have blue eyes, you're a mutant, just like me. Okay, another agent that can cause change, which would then change the allele frequencies, which would cause evolution, is migration or gene flow, movement of alleles between populations. Number three, agents that cause change in gene frequencies is genetic drift. So if you're drifting in a boat, you're not really going anywhere with any purpose, right? And genetic drift occurs more likely in a small population rather than a large population. So on your small population size can lead to genetic drift, changes in the allele frequencies of a gene pool due to chance events. Smaller gene pools are more impacted by drift. And the first example of drift I want to explain to you, and we're going to differentiate between the two, is the bottleneck effect. So here you can see all the different colors in this bottle. When I invert this bottle for a short amount of time, only a small amount is able to get through that crisis that occurs. This does not represent the whole. And so this crisis, this bottleneck, is limiting how many can get through. So this is not representative of our original population. There's some kind of weird yellow color. I don't have that now in my smaller population. Where might you see this? Let's say you have a population of frogs, right? And it's a pretty small population, 1, 2, 3, 4, 5, 6, 11 frogs, and a tree falls. And it just so happens to wipe out all the brown frogs when it falls. So this small population now is changed forevermore. They are only green from here on. It doesn't mean the green ones are more adaptive. This doesn't have anything to do with adaptation. It's just chance, and chance has a bigger effect on a smaller population. So underneath bottleneck effect, I want you to put a disaster. A bottleneck effect is a disaster. Okay, another form of genetic drift is called the founder's effect. Now, the founder's effect is an example of when you see this in the Amish, and I think it's like the Pennsylvania Dutch, but I'm not 100% sure. The Amish came when they were trying to have within their religious freedom, they wanted to practice what they wanted to do, so they came here to the United States. Just so happens on the boat that carried this one group of Amish, which tend to just breed with other Amish, there happened to be a six-fingered dwarf. And as a result, you can see a higher degree of having 6 fingers, okay, within their population and dwarfism than in the general population, because they founded a new population. Within that, they had that mutation, and because there was inbreeding within that population, now you have this trait at a higher frequency. So founder effect is a portion of the population starts a new population with a fraction of the total alleles, And then here, I already showed you that. This would be in a form of genetic drift. So this particular deer is not very popular. Why doesn't anyone love me? Then there is a forest fire, and it wipes out a big population. Now, all of a sudden, he's become very popular because the population has gotten smaller, and you're going to see an increase in this coloration of deer, not because it was best adapted, but because it was a small population, and chance eliminated a lot of the other alleles. Okay, and then another agent that causes change is non-random mating. Non-random. Remember, random mating would be anyone will do. Random mating, non-random mating is when you're purposely picking. You're the largest, the strongest, the best food, the prettiest feathers. Okay, you are choosing an organism on purpose to mate with. So non-random mating, one mate is better than another. One mate is better than another. And then the last one is selection. Okay, these flowers are more likely to get pollinated if they're prettier, if they're pollinated by an insect or an animal, if they smell better, that's going to draw them in. This hummingbird is more likely to be successful if it has a long beak because it's able to get at those nutrients. That is a form of selection. It leads to adaptations. So when we look at, here's The Origin of Species by one of the original copies by Charles Darwin. I just thought I'd show you a picture. Okay, what is it saying? It's saying evolution is the cumulative changes in groups of organisms through time. Populations evolve, not individuals. So this theory states that A, there are variations in a population, Inheritance of these variations would be genetic, it's in the genes. Some variations are more adaptive than others. And better adapted organisms produce more offspring, and there are vast amounts of time. So all of these things, right? Mutation, migration, non-random mating, natural selection, small populations, those will all contribute to change over time in the allele frequency, and that is evolution. So a quick review on what adaptations are. For instance, here, if this is what Darwin would say, that the reason why giraffes' necks got longer over time is because you would have variation amongst the population of giraffes, but those who had the longest neck were able to get the leaves at the tallest trees to be able to become bigger and stronger and more likely to mate. That would be a trait that would help them to survive and reproduce. So an adaptation is defined as a trait that helps an organism be more suited to its environment, And when the environment changes, there can be different results, right, from an environment changing over time. So, for instance, the amount of blubber, right, that you have. If the sea's warm, right, which would be bad on so many different levels, right, maybe they would have less blubber because right now, blubber is an adaptation that leads to this success. Also that their lungs can compress under pressure. All of these are different adaptations that help this organism to survive. Here, when the environment changes, which is a fear, three different results. You can get an increase in some numbers of the species who are better adapted to that particular change. You could have an emergence of a new species that is better adapted, or you could have extinction. So when environment changes, there may be three different results. Increase in some numbers of a species, emergence of a new species, that would be speciation, and extinction, which is probably most likely. All right, and that's where we're going to stop with this video. I will continue on and finish out this video in another section, okay? But so I'm going to put a pause. This will be on Edpuzzle, but I'm going to finish out right now for my YouTube channel. So we'll pause here for the Edpuzzle, but I'm going to keep going here for to make one video on YouTube. All right, so part 3 then is speciation. What leads to speciation? And this part we'll go through pretty quickly here. So speciation is change over time to where one species will not interbreed with another. So on your notes, speciation is a form of macroevolution, and you have that. This is 27.4, formation of a new species. All right, so there are different ways this can occur. Oh, someone's coming to my door, so I'm going to pause just for a minute here. Okay, I'm back. So the most common form, I don't know where I left off, sorry about that. The most common form of speciation is what's called allopatric speciation. So allopatric speciation starts off with some sort of geographical barrier. So here we have all these beetles, and there's some variations in the genes there. I can see some different colors in there. So then there's some barrier that separates this single population right here. And that barrier persists over time. Now, each population is going to start off with their own initial gene frequency, their own selection pressures for the environment they're in, and their own mutations that they have. So this one here obviously had a mutation where they have this green color. And so even if the barrier is removed, if they do not interbreed any longer, then this would be a form, then they have two different species. So it starts off initially with some sort of geographical barrier. So underneath your notes on 27.4, speciation, macroevolution, allopatric speciation is defined as species separated by a geographical barrier. And then they undergo different selection pressures. And those could be adapting to their environment on either side of that barrier. And they would also undergo different mutations. And they also have a different initial gene frequency. So you can add that all on there. Okay, under different selection pressures, they have different initial gene frequencies and different types of mutations. So let's look at an actual example of this. So here, these are salamanders starting off here in Northern California. As the salamanders migrated south, then they had Death Valley right here in the middle, which salamanders don't do well in. So they had to, when some went left and some went right. And over time, they had their different initial gene frequencies, different selection pressures, different mutations. They accrued changes. So even though when they finally migrated far enough where they did not have that desert separating them, they no longer would interbreed. Okay, so that's an example of the salamander species in California. And this is an example of what they call a ring species, because it forms a ring of variation. Sympatric speciation, less likely. They are literally side by side, and there is a mutation within one that then isolates it, reproductively isolates it from the other species. So an example, though, where this has happened is in Australia at Lord Howe Island. These trees were side by side, but there was a mutation in one that reproductively isolated it, so it no longer interbred with the others. So sympatric speciation is defined as speciation without a geographic barrier, usually involves a change in chromosome number. So here you can see two types of speciation. Could you identify which one is allopatric and which one is sympatric? Okay, hopefully you're seeing the change right here. There's a geographical barrier. So that would be allopatric. And here you have a mutation within that group, no geographical barrier. So that would be sympatric speciation. Okay, another form of speciation is what's called adaptive radiation. This is very common if you've heard of Darwin's finches, where there is maybe a founder effect like what we talked about earlier. So you have your initial species in an area, and then they're radiating out to explore every type of niche to reduce the amount of competition. So maybe some of the birds eat new leaves, some of the birds eat older leaves, some eat larger seeds, some eat smaller seeds. Why can they do that? Because they have mutations within their species that some had larger beaks that could handle the larger seeds and some smaller beaks, and some could even use a tool to get insects out of the tree. So adaptive radiation is a single ancestral species gives rise to a variety of new species. Think Darwin's finches or what's called the honey creepers. So here I've stacked some honey creeper heads up for you right here. You can look at the variations in the beak. And this one is an even larger radiation that has 20 different species coming from an ancestral species on the Hawaiian Islands. So their beaks are specialized for their different types of food. All right, so now let's talk about the pace of speciation. So the pace is how quickly it happens. So there's two different ones. There's what's called punctuated equilibrium. And I think of punctuated as very rapid evolution. And by rapid, and you look at the geologic time scale, it's over a course of like 1,000,000 years. Okay, that's considered very rapid. And there's the gradualistic model, which means it takes place over longer amounts of time. So here we can see here, this is the gradual model, gradualism. So it's slowly accruing differences, and at some point, there's no longer interbreeding. And punctuated equilibrium is there was some fast or rapid mutation that caused that reproductive isolation. So pace is speciation. Gradualistic model, speciation occurs after populations become isolated, each group continuing slowly on its evolutionary path. And you would have a lot of transitional links between it. And then the punctuated equilibrium model, species can appear quite suddenly, punctuated. I think like it's not punch, but I think like a punch is fast, so punctuated kind of sounds like that. So quite suddenly, and followed by a period of equilibrium or stasis. And it could be, those are the two extremes, really long time, really fast, and it could be anywhere in between. So it could be both depending on how quickly the environment changes. All right, so here's another diagram showing you phyletic gradualism with a lot of intermediate, and then punctuated equilibrium. You would not see a lot of those transitional forms. All right, so that is it for us on evolution. Keep in mind the big picture, right? First of all, gave you evidence for evolution. Then we talked about the process of evolution, which is a change in allele frequencies over time. And then we talked about how that macroevolution could come about, both allopatric, sympatric, or adaptive radiation, with of those, allopatric speciation, a barrier being most likely to occur to initiate that speciation, and then the speed of it. All right. I hope you are having a great day, and I'll see you in class.